All right, dual credit biology students, here we are. We're back for Chapter 4, Carbon and the Molecular Diversity of Life. In this chapter, we're going to talk a little bit about carbon and why carbon is such an important part of living organisms and why carbon is so friendly or so able and easily bound to other elements and bound to other atoms to create these complex molecules that are necessary for life. We're slowly working our way up to actual living creatures. You'll notice, remember that idea of reductionism, where we break things down into their little parts? Well, we're slowly starting to build things back together again. We started out by talking about individual atoms, and now we're getting to the point where we're building more complex molecules, and eventually those complex molecules will end up in living creatures. So we're getting there. I hope everyone had a good holiday weekend. I have no idea why a 3-day weekend goes by so fast, but 3 days in school seems to drag by. I think it has something to do with Einstein's theory of relativity, but perhaps we'll check that out in a physics class later on. So here we are, Chapter 4, Carbon and the Molecular Diversity of Life. Let's go ahead and get started, and we'll break this up into a couple of sections like we always do. Just as a brief overview of the chapter, carbon is pretty much the most important element in living creatures. Living organisms consist mostly of carbon-based compounds, and the reason carbon is so important is because it has this amazing ability to form large, complex molecules that no other element really has the ability to form as many different types of complex structures and molecules as carbon does. So all of the materials that make up living creatures, proteins and DNA, carbohydrates, all the things that you would find primarily in living creatures as opposed to inorganic things like rocks, well, those are almost all composed of carbon compounds. And the reason carbon is so flexible, we're going to find out, is because of its chemical nature. The fact that it has these four outer electrons in its outer shell, and it can form 4 covalent bonds with 4 potentially different elements, and that gives it like a huge ability, a variety of ways to configure itself. So here in this picture, we have this lovely elk of some kind. I think your textbook says what kind of elk it is. Roosevelt elk, not familiar with that species. This looks like the upper northwest. This looks like around the Seattle area or the western coast of Oregon or Washington, based on just what I'm seeing in the picture there. But all of the living creatures, whether it's the moss or the ferns or the grass or the trees or the elk that you see, including the ones you don't see, like the bacteria that live in the elk's gut, they are composed primarily of carbon-based molecules. So that's where we're going to take a look. Concept 4.1 is that organic chemistry is a study of carbon compounds. That word organic, you've probably heard it many times. If you watch TV for more than 30 minutes, you're bound to see a commercial where somebody is touting something that's likely to be called organic or all natural or healthy in some way. The word organic may mean a lot of different things in TV ads and health food stores, but in science, the word organic means basically that it contains carbon, or more likely, usually carbon and hydrogen. But most basically, you could say if it's organic, it contains the compound or the element carbon, and it's one of these complex compounds, usually with carbon as a backbone or a base. So no matter what organic might mean in everyday language or on TV, here in science class, it's going to mean a compound that contains carbon. And these organic compounds, they can be pretty simple. Like, for example, methane, that's the natural gas that you and cows and other living creatures emit, usually from their digestive tract. That methane is a fairly simple compound of CH4. It's 1 carbon sharing covalent bonds with 4 hydrogens. That's probably the most simple of these types of molecules. And it can also range up to enormous molecules that are made up of hundreds of different atoms. So there's quite a bit of a range. Most organic compounds contain hydrogen in addition to carbon. Not necessarily all, but the vast majority of the ones we're going to look at contain carbon as a base and then hydrogen plus other elements. I like how your book does generally a good job of kind of telling you a little bit of the history of what we're studying. So I'm very hopeful that everyone's going to get a textbook here pretty soon, or at least some of us are going to be able to share some textbooks. Within the textbook, it kind of talks about this history of organic compounds and how even in the early 1800s, scientists were able to synthesize or create compounds in the laboratory. But one of the types of compounds they didn't really have the ability to create way back then in the 1800s or prior to that were these organic compounds. So a lot of scientists and a lot of people in general had this idea or this thought that was called vitalism. And vitalism was this idea that outside of chemistry and nature, that there was some sort of a life force that existed that didn't necessarily fall within the bounds of physical and chemical laws and science. And that without this life force, you couldn't get these organic compounds. So these organic compounds could only come from living creatures that had this life force, this invisible material or force that was difficult to describe and explain. So that was kind of the basic idea until probably about the early 1800s, 1820s, 1830s. And at that point, scientists actually began to do experiments where they were able to create some of these organic compounds strictly in a laboratory using chemicals with no living creatures involved. So once they were able to actually start creating these compounds, this idea of vitalism started to lose favor and give way to this idea of mechanism. And mechanism is the view that all natural phenomena are governed by physical and chemical laws. And by far, I mean, I would say pretty much virtually all scientists or scientifically minded people nowadays, when it comes to chemistry and those types of things, would definitely say that mechanism is the view that they hold, that natural phenomena are controlled by physical and chemical laws, and through the scientific method we can kind of discover what some of those laws are. Interestingly enough, the first organic compound that was created in the laboratory that kind of caused this move away from vitalism was actually urea. You may recognize that word. Urea is the material that's in your urine, in your pee. Urea gives off or releases, degrades into ammonia, and that's what gives your urine its smell. So when the scientists first created this urea in the laboratory, he was kind of shocked because he knew this material was an organic compound that came from living creatures. And so to make it without a living creature, just to make it using chemicals in flasks in a laboratory, was quite a surprise way back in the 1800s. So vitalism, again, is that idea that there existed a life force that was required to create these organic compounds. And then most scientists moved away from that idea once it was realized that we could create these complex organic compounds just chemically without the need to have a living creature there. If you study chemistry or biochemistry for any length of time, you're going to come across this famous experiment, Stanley Miller's classic experiment. It has to do with the origin of life on Earth. Of course, the origin of life on Earth is still a bit of a mystery. That's good. Without mystery, there would be no point in science. I mean, if we had all of the answers, science would cease to exist. So the fact that we don't quite know exactly how life first occurred on this planet is something I appreciate. It doesn't mean we will not find the answer. It just means we don't have the answer right now. But in the pursuit of this answer, Stanley Miller in the 50s, 1953 I think to be exact, did this experiment where he demonstrated that some fairly complex organic compounds that would be the initial ingredients for simple life could be created abiotically without other living creatures. So what he showed was there are certain instances in environments where complex carbon-based molecules will be created chemically or abiotically synthesized. And so the way he did this was he set up an experiment that is, to the best of his ability and to the best of the scientific knowledge that we had, kind of formed the conditions of early Earth. And so in this flask down here at the bottom underneath the Bunsen burner, he had a water mixture that was supposedly similar to the ocean water of early Earth, and he heated that up, just like the sun heats up the ocean nowadays, causes some evaporation. This evaporation allowed water vapor to travel up the tube into this second flask area. In the second flask area, there was a mixture of gases, hydrogen and ammonia and methane and other gases that would have been similar to the atmosphere of early Earth. And then he introduced electricity, a form of energy, by inserting electrodes in here and firing those off, somewhat to simulate perhaps lightning or some other input of energy which could naturally occur. And then, as that material condensed and cooled, water droplets with molecules in them would form and travel down and eventually would land in this beaker. So this beaker was like the final holding pen for the samples that then they would chemically analyze. And within this beaker, they found organic molecules that indeed are some of the basic building blocks for living cells, living creatures. And then later, actually, in the 2000s, another scientist went back and using some of the same samples and some of the same experimentation method, of course, now we have much better testing methods than they did in the 1950s. They actually retested some of this material and retested new material that they made, and they found not only the same large organic molecules, but some additional large organic molecules that Miller didn't find the first time around. All right, so on to Concept 4.2. Concept 4.2 says that carbon atoms can form diverse molecules by bonding to 4 other atoms. And this gets into the fact that carbon, because of its atomic makeup, because it has those 4 valence electrons, has the ability to make a huge number of connections with other atoms. So it's the electron configuration that is the key to how the atom is going to behave and act. And the electron configuration determines what type of bonds, how many bonds that particular atom can make. And carbon just happens to have excellent configuration to make a lot of different bonds. I like to think of atoms and the way that they stick together to form molecules, kind of like Legos. My son and even myself like to play with Legos. And if you think of atoms as Lego bricks, well then carbon is kind of like the ultimate Lego brick. It can make more connections and have a more a larger variety of connections than any other Lego brick in your pile. So if you consider that atoms put together in certain arrangements make molecules, and then molecules put together in certain arrangements make cells and human bodies and deer and all the living creatures you see. Well, carbon is like the ultimate Lego brick. With it, you can make more different models than any other type of brick. It's kind of difficult to think about the fact that the actual shape of the molecules makes any big difference, because we're talking about things that are so small that we can't see them with our own eyes. As a matter of fact, except for some of the larger molecules, are even difficult to see with some of the most powerful microscopes we have. But the shape of the molecule does make a huge difference. And carbon can make a variety of differently shaped molecules. If the molecule has multiple carbons, and each carbon can be bonded to other atoms, well, they're going to take what's called a tetrahedral shape, and I'll show you a picture in just a second. If the carbons are joined by a double bond, like a double covalent bond, that means they're sharing 2 electrons instead of just sharing 1 electron. Well, then they're going to be in a longer plane. In other words, the atoms are going to not form this 3D tetrahedral, but they're going to be in the same plane. A picture is always better when you're talking about something like this. So here's that methane gas we were talking about, Chp4, probably one of the most simple carbon compounds there are. There is carbon with 4 hydrogens attached. Here's a structural formula, just a line drawing. And then here we have a nice ball and stick model. And look how it kind of forms this sort of pyramidal type shape. Tetrahedral is what it's called. And then here's a 3D space filling model. And then we have a model here, slightly different shape. We have ethane, and again you can see the structural formula, and this is like 2 overlapping tetrahedrals. Again, you see the single bonds where carbon is bonded to 4 other things, just like up here carbon is bonded to 4 other things. We have 2 of them here. They're overlapping tetrahedrals. But then we said if the carbon is double bonded, like here we have these 2 carbons are sharing 2 electrons, and they're bonded each to individually hydrogen atoms. Well, in that case, they're all going to be in the same plane. In other words, they're more 2D shaped, like you see here. They're all in the same plane. This is representing that double bond there, and this would be the 3D model. So it's the electron configuration of carbon, the fact that it has the ability to make these 4 bonds that gives it its ability to make so many different types of molecules. The valence of carbon and its most frequent partners, hydrogen, oxygen, and nitrogen, that's what makes up the basics, the building blocks that then are built off of to build complex living creatures like trees and whales and human beings. Here's a quick picture that shows the different valences of these major organic molecules. Of course, hydrogen only has a single electron, so it would do 1 covalent bond to try to fill that. Oxygen has 2 empty spaces basically here. It has 6 in its outer shell, so it can make a double bond, or it can make 2 different covalent bonds. Nitrogen has 3, and then carbon has the 4 that we're talking about here. So carbon can easily bond 4 covalent bonds with 4 different other elements, or another carbon, or perhaps a double bond with a carbon here and a hydrogen here. And there's almost an unlimited possibility as far as the different combinations can be. While hydrogen is probably one of carbon's most prolific partners, like you see carbon and hydrogen together an extreme number of times when you're looking at these organic molecules. Carbon can bond with other things, especially the ones that we just looked at, oxygen, nitrogen, things like that. So here we have the carbon dioxide molecule, just a line drawing, and you see carbon dioxide is carbon sharing a double bond, double covalent bond. That's what the 2 lines mean, with 2 different oxygens. And you notice they're kind of in the same plane there because of those double bonds. Down here, this little bit more complex molecule on the right, this is urea. The urea molecule we talked about, that was actually that first molecule that was discovered, that was made by man just in a laboratory without the living creatures involved. And it's an organic molecule. This is what you find in your urine. It does break down into ammonia and give it that smell. So you can see how the carbon can bond to oxygen, the carbon can bond to nitrogen. We already know it can bond to hydrogen. So there's a lot of different possibilities there. Since carbon can also bond to itself and form these chains, a lot of times you see these molecules that are in a long chain-like form. The carbon chain kind of forms the backbone or the skeleton of most of these molecules. And the carbon chain can be of a huge variety of shapes and lengths and different configurations. Here we have a little animation. It won't play in the podcast here, but if you go to the PowerPoint notes, or I think to the PDF notes, you can take a look at this quick animation about carbon skeletons, and then I'll put the file online, to the animation file online separately, so you can find it on the website. If you didn't watch the animation, it goes on to kind of talk about some of the different ways that these carbon chains can be formed. So one of the ways that can be different is the length of the chain. So here on the upper left, where it talks about length, we have a fairly short chain of only 2 carbons. Here's a longer chain, propane, of 3 carbons. This is what you burn in your barbecue grill. But these chains can grow to tremendous lengths. There can be many, many, many carbons in a long chain with a wide variety of molecules, a wide variety of atoms, whether it's hydrogen or others branching off of it. Also, let's see, we'll go down to B. We can have different forms of branching. So here we have butane, which is another type of gas that some homes use butane to heat the house or to cook on. So here you have butane, which is really only different from propane in the fact that it has an additional carbon and some additional hydrogen. And then we can have a different type of molecule that may actually have different styles of branching. So instead of a straight line, we may have 90-degree angles and different types of branching. And this actually affects the molecular shape. So although that's not something we can see with our eyes, we're going to find out that the actual shape of the molecule has a huge effect on the way it behaves and how it affects cells and the living parts of our bodies. And then let's see, up here on C, we have double bond position. Here you can have changes in where the double bonds are located. So if you look at this molecule here, you can tell that the double bond is located between the last 2 carbons here. And here the double bond is located in the center of the molecule. Even something like that can make a difference, the double bond position. Last but not least, carbon can do more than just form straight lines. Carbon has the ability to form rings, like you see here. And so carbon can bond to itself in a ring shape and then have other atoms kind of bonding off of the outside. And here we have a ring where there are some double bonds between some of the carbons and single bonds between some of the carbons, and then other atoms, this one, hydrogen, off to the side there. I said that hydrogen and carbon are perhaps the 2 that are most commonly linked together, and we have a name for these. They're called hydrocarbons, which kind of makes sense. The breakdown of the word would tell you that it's made up of hydrogen and carbon, and these are organic molecules that only have those 2, carbon and hydrogen. And there's a lot of organic molecules that have, at least as part of their structure, something that we would call hydrocarbons. Like fats have a part of the fat that is a hydrocarbon component. And hydrocarbons are so important because the bonds between the hydrogen and carbon can be broken and formed and undergo reactions that can release a large amount of energy or can be used to store a large amount of energy. So, for example, fat in the human body. This picture here is a microscope picture of some fat, human adipose tissue, adipose cell. There's the nucleus of the cell. So we're really zoomed in very close, and you don't get to see the entirety of the cell, but you're only looking at a portion. This is droplets of fat that are contained within that cell. So in your body, in your lips, or around your waist, or wherever you might have deposits of fat, you're going to have cells that look like this with fat droplets contained inside. And if we could zoom in even closer, we would see the individual fat molecules, and we have carbon and hydrogen components here, hydrocarbons. Fats have these tails that you see here of hydrocarbons. So in the yellow portion, in the highlighted portion, those are hydrocarbons. Last part of this section we're going to take a look at before we take a break is something called isomers. And again, it's interesting to think that the actual shape of the molecule really makes that much difference. You would think that as long as you know the elements that make up that compound, that's all you would need to know. But in truth, the three-dimensional shape of the molecular compound does make a big difference. So isomers are compounds that have the same molecular formula, but they're shaped differently. They have a different structure that also gives them different properties. And we're going to look at three different ones. We're going to look at what's called structural isomers. And structural isomers have different covalent arrangements in their atoms. In other words, they have the same formula. They'll have the same number of the same types of atoms, but they'll be arranged differently. They'll be in a different shape. And I'll show you a picture, of course, in just a second. The other type of isomer we're going to look at are called cis-trans isomers. And cis-trans isomers are unique because they have a different spatial arrangement. And again, that just means they are differently shaped in the way they're laid out and where the particular atoms are. So we're going to show a picture of that as well. And then the last ones are called enantiomers. And enantiomers are isomers that are mirror images of each other. Again, they have the same formula. There's the same number of atoms or particular elements, but the way they're shaped means there's almost like a left-hand and a right-hand version. And again, these words, very difficult without pictures. So let's take a look at a picture. This is actually an animation. So again, in the PDF or the PowerPoint, click on this and right-click and play and take a look at the animation. At the top, we have structural isomers. So here you see we kind of have a chain. It kind of looks like a stair-step chain. And then here, if you count the balls of the different colors, which represent atoms and particular elements, you'll find that they are the same, like the carbon 1, 2, 3, 4. 1, 2, 3, 4 are the black balls, which represent carbon. But you'll notice this is kind of in a T shape. And this is more in a stair-step shape. So if you counted the number of elements, you'll find they're the same, but the way they're put together, it has a different shape, a different structure, and that actually has a big effect. The second one here in the video is called geometric isomers. That's a term that a lot of times we don't use anymore, but just remember that if you see this word geometric isomers, that is what we call a cis-trans isomer. Cis-trans isomer is geometric isomer. And then the last one here is enantiomers, which I have a horrible time pronouncing, enantiomers, and those are the ones that are mirror images of each other. So click on the link to animate and take a look at this, and we'll go on to the next slide and I'll kind of discuss it a little bit further. Alright, so just a little bit further discussion on isomers. Again, isomers are compounds that have the same molecular formula, but they have different structures. They have different three-dimensional shapes because of the way that they are put together. So at the top we have structural isomers. Again, here you have two different compounds, or two different isomers that have the same number of hydrogens and the same number of carbons. So we have 1, 2, 3, 4, 5 carbons. 1, 2, 3, 4, 5 carbons. But notice the way they're built. This one's in a long, thin, direct line. This one's in a 90-degree sort of T shape or L shape. That's a structural isomer. Down here in the middle, we have what's called cis-trans isomers, which is an odd-sounding name. But the cis prefix is Latin for meaning on this side or on the same side. So let's say you have two isomers here of a particular compound. And in this isomer, in the cis isomer, X is just representative of whatever element you want to put in there. It doesn't matter. So X is just kind of a variable that we're sticking in there. Notice that the X is located on the same side. So that would be a cis isomer. And then we go to the next one here. It's the same compound, but notice how the X's are not on the same side. This is called a trans isomer, and trans, that prefix in Latin means across. So they are across from each other. So a cis isomer means they're on the same side, whatever element you're talking about, whatever atom you're talking about. And then the trans isomer means they are on the opposite side or across from each other. And the reason, you might think, well, what if this just twists? Like if this right-hand side, or actually this left-hand side, if it were just to twist down, it would be the same as this one. And that can actually happen if it's a single bond. But in a double bond right here, that double bond prevents that. So these two will be different from each other because there is no rotating around that double bond. So that double bond locks them into place either on the same side, as in a cis isomer, or on opposite sides, as in a trans isomer. And then the last one down here, the one that's hard to pronounce, the enantiomers. Told you it was hard to pronounce. Enantiomers. The enantiomers are like a left and right isomer. So, for example, here you have a molecule, a compound that again has the same chemical formula, but the way it's shaped, they are like mirror images of each other. And that's kind of the equivalent if you think about, like, for example, you have gloves, a left-hand glove and a right-hand glove, and your hands and the gloves are mirror images of each other. Your left-hand glove won't really fit on your right hand. So even though they are the same layout, they are mirrored of each other, and that does have a big effect. As a matter of fact, that has a very cool effect when it comes to talking about drug use, as far as medical use for different drugs and things like that. These enantiomers are pretty important in the pharmaceutical industry because it turns out that you can have a drug that's made up of a certain compound, and it'll have different enantiomers, a left and a right, and almost always, only often, one of these versions is effective and one of them is not. So usually only one of these isomers is biologically active. And it's pretty interesting to think that you can have the same molecule, the same compound, and it won't have the same effect just because of its 3D arrangement. So that demonstrates how sensitive that living creatures are just to these slight changes in the way that these molecules are put together. Here we have a quick animation about a particular compound called L-DOPA. And so if you'll right-click on this and hit play, it'll talk about how these enantiomers, how they make a difference, how the left one might be active, but the right one is not within the human body. And then here's a good example if you didn't happen to watch the video or you just need one more example. The drug ibuprofen that you might take just if you have a headache or sore muscles or something like that, it has two enantiomers. It has what's called an S and an R enantiomer. And here are the two models of them. The one on the left here, the S ibuprofen, is effective. It's what will help cure your headache or help relieve your sore muscles. But the R ibuprofen is the same molecule as far as the formula. It has no effect. It doesn't do the job. And then if you're familiar with albuterol, albuterol is a chemical that's used for asthma sufferers. It helps shrink inflamed passageways in the lungs and help reduce asthma symptoms. So albuterol is the same way. Again, it has two enantiomers, an R and an S is how they define them. The effective one is this one, the R albuterol. The S albuterol has no effect or does not have the intended effect. So it's pretty amazing that the 3D arrangement can actually play a major part in the biological effectiveness of these molecules. All right, team, let's take a break right there. If I've given you a headache, go take some ibuprofen. Make sure it's the correct enantiomer. Make sure it's the S ibuprofen, because if you take the R ibuprofen, it's not going to do you any good. Go take a break, check your email, something like that, and come back, and we'll finish up with Concept 4.3.